Hi. So you know how your Apple Watches tell you to breathe? Just breathe. So this is going to be what my basic science friends will refer to as a soft talk. And I'm going to talk about some of the issues that we encountered as we have gotten an RA trial about close to enrollment and what the design is, what design factors we're taking into account as we actually design a cell therapy trial for children with refractory systemic onset JIA. And I won't be any more specific than that because that'll be, I think, one of the things with discussion. And I've been told I have between 6 and 7 minutes, and then we go to a panel. ARTHRITIS is a company in Cleveland that we've collaborated, not directly on the stuff I'm showing you today. So I think that many of the audience has heard about rationale for using MSCs. A number of my colleagues in the audience, I think, remain somewhat skeptical, and I hope that over time we'll show them that we're right, that it may be of value. To discuss the use of placebos in phase 1 safety trials in children and discuss what our choice as a community should be as we have the opportunity both to plan this clinical trial that's funded by the Arthritis Foundation and to implement it in year 3. And to help people in the audience and everybody who's a stakeholder understand what we actually have to go through when we're an investigator sponsor. And then also think about the way of easing some of the regulatory burden because that's definitely a deterrent to investigators and institutions sponsoring their own phase 1 studies. So MSCs are cells that we refer to as mesenchymal stem cells, but they may not, in fact, have their stemness or their ability to differentiate into cartilage, skin, and other tissues. That may not be their greatest function, but rather they think of them as medicinal cells, which deliver chemokines, cytokines, factors in vivo, where they both sense and then respond to their environment. So unlike a drug that has target, for different conditions, they may have the ability to respond differently. We're interested in them for their ability to tune down the immune response, allowing for host reparative mechanisms to act. So we really think of them as a hit-and-run phenomena, especially when they're allogeneic rather than autologous. So they don't stick around. So allogeneic is coming from somebody else. Autologous is coming from yourself. And so the mechanisms to protect the host immune system means that we're interested as a host in destroying cells that are foreign that come to us. So they appear to go to sites of injury and inflammation, exert their effect, but not stick around, but rather induce the host to repair whatever the tissue is. They're not recognized typically by the immune system as blatantly foreign because they have low levels of class 1 and class 2 MHC, so the transplant antigens. They lack what we call co-stimulatory molecules of CD80 and CD86, and so the pro-inflammatory signals that are transduced through CD28 on T cells are downregulated, and so they are largely invisible to the recipient immune system, except they probably, in an allogeneic fashion, get targeted with complement, which results in their decay. So everybody should memorize the slide, and then we'll move on. So this is a slide that Arnie Kaplan, who really got me started on much of this work, and I published in 2011, and there are about 60 more mechanisms of how MSCs might work that I could add. If your favorite mechanism isn't up there, please don't be offended. But these cells act at a variety of levels. They have the capacity to modify T regulatory cells, Th17 cells, B cells, dendritic cells, and so in some conditions like cancer, certain cancers, you may not want them because it may make you more susceptible to the cancer. And we're learning things about cancer that may be different from autoimmune disease. This is a paper published by Gary Gilkison and his group. He's very interested in using mesenchymal stem cells in lupus. And essentially, he's showing that activated mesenchymal stem cells, and they're activated by the local environment that they're infused into in vivo, they become activated, and then there's a lot of crosstalk between T effector cells, or the T cells that we think about as proliferating and maybe doing harm, T regulatory cells, which we think of as if they're active, may help downregulate the immune response, but also they may tune antigen-presenting cells so that T cells do or don't recognize what they're showing to the T cell on their surface. And so this is a summary diagram that basically is simplistically showing that we're shutting down some of the effector T cell functions that we think are important in perpetuating many of the arthritis diseases that we're interested in. And some of those functions are likely mediated by things like IDO, which can be anti-inflammatory, improving function, if not number, of regulatory T cells. The IDO pathway is, it's not clear if it's the metabolites of tryptophan that are metabolized that are the bad actors, or if it's increasing levels. So we don't know if it's the effect of IDO is negative or positive, or some of both. But we do know that at least in vitro, we can shut down those responses. And I'll show you what are the kinds of things that we look at ex vivo to know whether or not these are cells that we're interested in. We know from the literature and from the published experience that in graft versus host disease, when people have bone marrow ablation, they receive somebody else's marrow, that using MSCs or infusing MSCs reduces graft versus host disease both in a therapeutic manner and in a preventative manner. And unlike hematopoietic stem cell transplant, where radiation or chemotherapy is used to get the person ready for a bone marrow transplant, we don't need to precondition these people when they're receiving MSCs. We essentially give them acetaminophen and sometimes Benadryl. They do send some repair local damage, and we don't think that they're globally immune suppressive. So that perhaps we think about the biologicals often that we're paying a price for our ability to shut down abnormal responses, that there's some impairment of host responses or vaccine responses, so you may be able to fight off infection less well or respond to vaccines less well. We don't think these are globally immunosuppressive. And so our hypothesis when we started this journey in rheumatoid arthritis, and now we've expanded it to systemic onset JIA, is that we can induce immune tolerance. And at least in early RA, we think we might be able to do an immune reset. But the question is, can we do the same thing in refractory systemic onset JIA patients? We're obviously not going to be able to reverse everything, but can we help the immune system stabilize so that the host reparative mechanisms have a way to restore tolerance? And we know after giving MSCs, we need to know in these trials, because early clinical trials in a phase 1, they're done for safety. But the secret that nobody's going to tell you is everybody at least wants to see a little efficacy because they don't know whether or not to pursue it. So we say they're done for safety, but what we really want is some sort of biological signal, if not clinical signal, to show us that we're on the right track and we're not going to spend 10 years going down a wrong pathway. So this is one of the assays we use in our lab, and this is essentially pretty simple. We load up the cells with a red dye. And so we give stimuli in the laboratory that mimic what a T cell might see, and we ask whether or not the T cell divides. And so shown on the left is a normal amount of divisions of T cells that we'd see over about 5 days. When we use products from mesenchymal stem cells that we've tweaked with cytokines, we can show that we have a single peak and almost none of the cells divide. And so that we've used to pre-qualify some of our cells, but obviously we need better potency assays when we go into later clinical trials. And so part of what we need to understand is what should a potency assay be in systemic onset JIA? So what means that those cells are actually going to be effective? Because the answer for rheumatoid arthritis may not be the same answer as for systemic onset JIA, and may not be the same for a third-degree burn. So can we personalize those MSCs, not necessarily to the individual, but more to the condition to induce the response that we want to see in the person receiving them? So everybody always asks, are MSCs safe? And now there's safety data in a myriad of diseases. If you talk to your transplant colleagues, they sort of see it as not much different as giving a dose of steroids and perhaps less harmful. I think the rheumatologists aren't quite there yet. It's been done in Crohn's, graft versus host, multiple sclerosis. There was an autologous trial excuse me, by Jeff Cohn in Cleveland, and he's now going into phase 2. He had Department of Defense money to try and look at relapsing and remitting multiple sclerosis and see if he could slow down lesions. And the cells were safe, and now they're going into phase 2 for efficacy. And there's been some thought in diabetes. Obviously, diabetes appears to have a long lead-in time, so you may need to do it much earlier in what would be called preclinical disease. And I talked about infusional toxicities being pretty minimal, mostly fever. At least there's no tumor there's not tumor formation in the studies that have been observed in the U.S. I don't know how many of you saw the New York Times about 9 to 12 months ago where there was a tourism study about somebody who got a tumor somewhere in Western Europe in their spine from getting MSCs. And so as we proposed this sJIA trial, I felt like we were in a position to own this therapy and try and understand if it really worked and was of value rather than losing data when people go into a non-study situation. Okay, some safety references for your reference. This was a trial that was presented at EULAR, and this company used mesenchymal precursor cells, which are slightly they're early MSCs. And what I really wanted to point out is that while we didn't see any ACR70s in the placebo group, even in a very early phase trial with the MPCS at 2 million per kilo, we were able to see some effect. And the interesting thing is that you also have remarkable effects in the health assessment questionnaire, so that patient-reported outcomes, like health assessment questionnaire and pain, may change faster than the usual composite measures we're looking at. And I think that's important to keep in mind as we design trials. So this is our phase 1 that has gone back hopefully to the FDA for its final amendment. It's funded by NIAMS, and so we go back and forth between minor tweaks, and hopefully we're almost ready to enroll. The cells have to go out for what's called adventitious testing, so that they do a variety of viral testing. It takes about 6 weeks to do those. And that's a list of studies that use them. So issues in terms of particularly for sJIA, I think, for MSC therapy as we design this is what's the best clinical trial design for this? And should we be proposing, since we're going into refractory disease, should we propose single dose or should we propose multiple dose? Right now we're funded to do single dose. Just and his colleagues have included systemic onset JIA in their cohort of JIA who they're treating, and that's a multiple dose regimen. So I'm sure they'll have some input from us for us. We also need to, at least when we move to phase 2 or 3, develop potency assays. So what cells are okay to go into a person with what disease, and that may be different from disease to disease. The product may need to be looked different, but we've been told by the FDA for our RA trials when we move into a phase 3 that we will need to have a potency assay. So we're actively working on that in the laboratory. And we do need measures of biological response early on. We've chosen some for the early RA trial that are acceptable to both NIAMS and to the FDA, and we hope to detect a biological response after we deliver cells sooner than one might see a clinical response. So issues in sJIA have to do with, are we better off doing this in an autologous versus an allogeneic fashion? Autologous on the surface seems safer. However, if they're not going to be as effective, so in lupus we know that there are early defects in MSCs in lupus, and there's been some suggestion in rheumatoid arthritis that some of the MSCs may be prematurely senescent. When we do allogeneic cells, we typically use them from donors under 30 who are healthy. There are a variety of sources of MSCs, and so we are most used to growing up bone marrow transplant one. There are certainly companies that have both adipocyte and umbilical cell products, both national companies and local companies. And so I think the question is, if we know what we're looking for in an MSC to treat sJIA, then we can test all the cells in a particular assay to try and understand which might be best. And we might not be able to do that for the phase 1 as thoroughly as we would do once we're moving on. And so another design issue, I think, is should we be using placebo? I will tell you in the RA trial, the FDA asked us to use placebo patients. So there are 15 patients at 3 different doses and 5 placebo patients that will be enrolled. And the FDA asked us to do it, and we were reluctant, and the study section told us to do it, and we did it, and the study was subsequently funded. But I think this is going to be a question for our FDA colleagues, is under what should we, if we really want to get a good trial, should we be using placebos, and then what's going to be acceptable to parents and patients? Should we do a time to response phased clinical trial? If we should do that, how long an interval? Should we do time 0 and time 1 normal saline and 1 stem cells and be blinded to that? Or should we propose open label for all the patients? And I think that's an issue that we're grappling with, and we're grappling with it as we do some focus groups to try and build a trial that is both robust and people will want to enroll into. Along with, before I get to that, along with that is we have funding to do to enroll 12 patients, and so I think one of the questions is with refractory systemic onset JIA, do we want to enroll patients with systemic symptoms, patients with joint systems, some of both, and should we be prescriptive about how many of each, or should we just take anybody who meets inclusion criteria in the order that they do? One of the issues that you don't realize when you first start this or have never done this is all the regulatory issues that you get into, and it seems sensible to use electronic means to record data. When you use electronic means to record data, you have automatically incurred a level of expense that as an investigator you didn't anticipate. So there's something called Part 11 compliance, which really speaks to what are the regulations for assuring the safety and integrity of patients and the data. And so there are audit trails for what data you enter, who enters it, whose permissions. You have to monitor those audit trails, and you have to validate your database. These are really expensive things because none of these databases are completely open source. So industry tends to use Rave, and there's another Inform, and things like that. Those are very expensive for a small investigator-initiated trial to sponsor. We're using a database that's a spinoff of the University of Wisconsin, and I've learned a lot doing this, but the CF Foundation really got it right in the first place, and they built their own database out of this whole venture philanthropy model, and so that's available to their investigators. Some of the other institutes at NIH also have databases that when they fund a study, their investigators use that are Part 11 compliant. REDCap is a product out of Vanderbilt, and if your institution chooses to do those audit trails, you can use REDCap, and there's also a company that's trying to spin REDCap off where they host the Part 11 compliant piece of it. There are also things like CDISC, which we're not using. CDISC terminology is a terminology that the FDA likes industry to use so that all the data fields look the same and they're not getting 5 different names for a single data field. We have not used this in our RA study. There's something called metadata and HUDRUG, and these are for concomitant medications and coding adverse events. As you imagine, all of these come with license fees and are often beyond what the individual investigator can do when they're doing an early clinical phase. So benefits and problems. Okay, I will refer you to the slide. I think one of the questions is at any point will the FDA invite patient organizations to weigh in on this, or are there venture philanthropy models we could do to reduce the burden, because that, I think, would speed the process of investigator-initiated trials. Take-home messages, we think that cell therapy has promise, that it's expensive, it's still early in clinical development. We can't scale up the way we can with regular drugs, but I think the people in the room can help us understand what's an acceptable clinical trial design. There are obviously trade-offs, and whether or not can we approach the FDA about easing the Part 11 compliant regulations, or can we do some sort of venture philanthropy thing like the CF Foundation has done with all the technical help in the room to try and create something that's essentially open source. So none of this gets done in a vacuum, so I would like you to sort of journey this with me, and we're at the beginning of the journey.